I would like to thank the organizers for giving me an opportunity to present today. The research of my lab focuses on understanding cellular and molecular mechanisms of complex diseases using machine learning and data science methods. And we basically approach these complex challenges by the analysis of single-cell sequencing data and multi-omics data integration. For today, for the first half of my talk, I'm going to talk about two projects that are involving analysis of single-cell sequencing data. And for the second half of my talk, I will focus on a project on the multi-omics data integration. Tumor microenvironments consist of different cell types, including immune cells, fibroblasts, cells from the tissue of origin, and malignant cells. The cell-to-cell interaction and the composition of different cells in tumor microenvironment have an impact on different aspects of the disease, including the drug resistance and immunotherapy response. So basically, it is important to study tumor microenvironments. Traditionally, bulk sequencing technology is used to measure gene expression profiles of the tumor microenvironment. However, in the bulk sequencing, thousands of cells are put together and an average gene expression from all cells are measured. This technology at the bulk level, it doesn't have the resolution to identify gene expression profiles of the individual cells. And therefore, it cannot help us to fully understand the cell composition in the tumor microenvironment. In recent years, single-cell sequencing has advanced rapidly. Now we can measure gene expression profiles of the individual cells. And this needs to be followed by some computational methods. So basically, first, the tumor cells are dissociated, the live cells are selected, and the single-cell sequencing technology is applied to measure the expression of the genes at the single-cell level. And some computational methods like clustering is used in order to identify the cells that are similar and representing a cell population. Analysis of cancer single-cell RNA-seq data requires high-performance computational infrastructure and user expertise to build analysis pipelines and to tailor interpretation for the cancer biology. The number of algorithms and computational methods for the analysis of single-cell RNA-seq data has increased considerably in the past years. And while this provides a lot of opportunities for the users to pick from the computational methods and the packages of their choice, they are left with the difficult task of deciding which tools and parameters they should use. In collaboration with Dr. Pugh and Brutno at University Health Network, we are building an intuitive and scalable web portal for the standardized analysis and exploration of single-cell RNA-seq data from cancer studies. This is the project is called Crescent. A typical computational pipeline for the analysis of single-cell RNA-seq includes mapping reads, quality control, normalization, dimension reduction, cell clustering, cluster labeling, detection of differentially expressed genes, and usually followed by some visualization using t-SNE or UMAP. Crescent makes all of these tools available in a web browser. So the users don't need to have extensive bioinformatics expertise in order to access to high performance computing infrastructure. And also because the pipelines are end-to-end, they don't need to spend time learning how to integrate various independent tools in order to fully analyze the data. We have collected and put several public data sets in Crescent and also multiple public pre-configured pipelines and modules are available through the Crescent. So the user can basically use them and do the optimization and reanalysis of the data if they want to. Here I'm showing you one example of how the control menu on the Crescent Web Portal would look like. So this data is, this plot is based on the data from the human tumor associated T cells, which is a public data set. And each point corresponds to one cell. and they are different cells are colored based on the clustering outcome that after we use Crescent, we can obtain these different clusters. And the default here is the Surat. Basically, you can visualize different clusters, or if you pick a gene that is differentially expressed between the cells in one cluster against all other cells, then you can look at the expression of that gene. In this example, this is CMC1. And you can see that gene is highly expressed in cluster 3, but not as much in the other clusters. You can also use the Violin plot to see how this gene is expressed across different clusters. The other feature of Crescent is that you can basically use the metadata provided by the users in order to project them into the UMAP or t-SNP plot. And in this example, the metadata that is used is the cell labels. So you can see different cell populations colored here. Or alternatively, you can use the numeric variable, for example, gene expression from orthogonal assays in order to visualize them. Currently, we are working on the assessment of the performance of different clustering methods and cluster labeling approaches, as well as CNV methods, and we want to identify the best performing algorithms when they are applied to the cancer single-cell RNA-seq data. And this is an ongoing process, and we aim to implement them in Crescent. The second project related to single-cell sequencing that I'm going to present now is a database that we have recently developed at Western. It's called TME Explorer, a tumor microenvironment single-cell RNA-seq database and search tool. The number of cancer single-cell RNA-seq datasets that are generated in the past years has increased considerably. And most of these data sets are available through GEO as part of the publication. And therefore, we don't have a database, a comprehensive database that includes all of these data sets together in one place. Most of the existing databases for the single-cell RNA-seq data consist mostly normal tissue or non-cancer data and few cancer data sets. So we felt the need to develop a package, a database that is specific for the cancer data, so that users can easily locate and access tumor microenvironment single-cell RNA-seq datasets in one place. My students, Eric and Alan, worked on this project together, and the package that they've developed is available both in GitHub and Bioconductor. Here I'm showing you a table describing the kind of information that we can get as a metadata for different data sets. So you basically can get the publication information, the technology that is used to sequence the data, data type, type of the cancer, number of patients, cells, genes, and whether the gene signatures or cell type labels are available for different data sets. And all the data and metadata are kept in a consistent format across all data sets. Two main functions of TME Explorer include Query TME and Save TME. So Query TME is the search capability of TME Explorer. You can use it just to visualize the metadata similar to the table that I showed you in the previous slide, or you can set some criteria to select the data sets of interest and then do your own analysis. The other function is Save TME. So basically you can write the data sets of your interest into the disk, and this includes the expression table, cell type labels, and gene signatures. In this example, I show you how you can incorporate TME Explorer into your analysis of the single-cell sequencing data. So basically, you start by using the Query TME function and set your criteria. For example, if you are interested to look up the data sets for which the labels of cells are available, you set this is true. And also you look at the data sets for which we have the gene signatures available. So this provides you with a set of data sets with these criteria. And then you can look at different values or different data sets out of that. So you can get the gene expression, gene signature, and cell type annotation. Gene expressions, for instance, can be used in combination with the SURAT clustering to identify different clusters in the data. And if you look at the outcome of the clustering and combine that with the gene signature, using some cluster labeling method, you can annotate your cells and identify the labels of cell clusters. In other words, you can get the cell type labels. Also, if you are interested in comparing the cell type labeling approach with whatever the cell type annotation the original paper reported, you can use it for evaluate your results. Okay, now I would like to switch the gears and talk about the other set of the projects, which is focused on the multi-omics data integration. We are living in a time that new technology advancements have enabled us to generate multiple types of biological data. This includes genome data, epigenome data, transcriptome, and microbiome data, among many other data types. So basically, we are equipped with a lot of data that we can explore in order to understand biological mechanisms of the diseases. For many complex diseases, there are usually multiple factors that are relevant, and therefore a single data type is not informative enough to give us the full understanding of the disease mechanism. And therefore, combining multiple types of the data together is considered as a crucial step for the same reason. However, the key challenge here is that how we can combine different methods in a way, in an effective way, so that it provides a good insight into the disease mechanism. In this part, I'm going to tell you about a project where we developed a model to integrate genetics association data, epigenetic data, and gene expression data in order to understand gene regulatory mechanisms of complex diseases. We initially applied and tested that on 9 different autoimmune diseases, but this is a general model and can be applied to other types of the complex disorders. As you know, different people have different DNA sequence, but when we look at this at the population level, we see that the difference is only 1 out of 300 bases or so. These are called single nucleotide polymorphisms. In genome-wide association studies, people are divided into 2 groups, cases that are people with disease and controls that are people without the disease. And the allele frequency of different allele frequency of each SNP is considered, and they're compared against compared between cases and controls to see if there is a significant difference in the allele frequency between the 2 groups. So basically, for each SNP, you can get a p-value of a genetic association. And if you apply it to all the SNPs in the genome, then you may get a plot like this. So this is a plot from, this is a Manhattan plot for the schizophrenia. Here, each point corresponds to a SNP. Of course, you see a line rather than a point because there are so many SNPs close to each other. That's why you see a line. X-axis shows the position of the SNP on the chromosome, and Y-axis shows the minus log10 p-value of association. So if the SNP is highly associated to the disease, then you see a higher signal here. And if the SNP significance level is, the p-value is lower than 5 times 10 to the minus 8, you call the SNP as a GBUS SNP. Here you can see that hundreds of regions have association signal to the disease, and that's because this is a complex disease. Now, if you look at these association loci and ask this question that where these variants are located and what functions they may have, the first guess may be they are located on the gene bodies. However, several evidence shows that many of these SNPs are not located on the protein coding genes, but they are in fact enriched on regulatory DNA marked by DNase1 hypersensitive sites, or DHS in short. The fact that majority of disease-associated SNPs fall into regulatory DNA support a model where disease risk is driven by alteration to gene regulation rather than changing the coding sequence of the protein coding genes. However, the key challenge here is that how we can go from this global enrichment of SNPs on regulatory regions to pinpoint the specific regulatory elements and genes that drive risk to the disease. Let me explain this problem a little bit in more detail. So if we focus in one of the loci that have association signal, then you can see that there are multiple SNPs located in this region. And basically you may ask this question that which SNPs are likely to drive risk? We cannot go and pick the most associated SNP and consider that as causal because association shouldn't be confused with causality. So we need to find which SNPs are likely to drive risk to the disease. Also, given that there are hundreds of epigenetic regions located in one locus, you may want to identify the regulatory regions that drive risk to the disease. And given that tens of genes may be overlapping an association locus, the question is that which genes are dysregulated. So basically, you can see that a single data type cannot answer all of these questions. You may need to have genetic association data to answer the first question, having epigenetic data to answer the second question, and gene expression data to answer the third question. And in fact, we want to combine these different data sets together in order to have a full understanding of the disease mechanism. So the key here is that how we can develop a model that can effectively combine all of these data sets together. In order to answer this question, we developed a model that is called regulatory fine mapping, or RegFM in short. It's a three-step model to prioritize specific SNPs, regulatory regions, and genes that drive risk to the disease. I'm not going to go to the details of the math behind this. I just want to give you an overall idea of how it works. RegFM starts with GWAS data, and basically here I'm showing you an association locus. Each point corresponds to one SNP. X-axis shows the position of the SNP on chromosome. Y-axis shows the minus log 10 of the p-value of association. And basically, you can see multiple genes or regulatory regions can overlap these regions. We first identify a set of SNPs that are in high LD to the most that are in LD of higher than 0.1 with the most associated SNP. And then use Muller et al. Method that is published in Nature Genetics in 2012 to measure the posterior probability of association for each of these SNPs. Then we identified a 99% credible SNP set, which is a set of SNPs that together explain 99% of the posterior probability of association, meaning that it's 99% likely that we observe the causal SNP in this set if it is genotyped or at least imputed. As a second step, we overlap these credible SNPs with the regulatory sites and identify the DHS or regulatory regions that are likely to drive risk to a disease. And then by assuming that each of these regulatory sites have an influence on the expression of the genes that they control, we look for a correlation between the activity of the regulatory sites and expression of the genes to identify which genes are likely to be controlled. So here I'm telling you how we do that. This is just an example. So it's basically a cartoon. So here you can see the activity of different DNase I hypersensitive sites across different cell types. And as you can see, these sites become open or closed depending on the cell type. So the patterns of accessibility varies from cell type to cell type. Similarly, for the gene expression, we can see the expression of genes can become high or low depending on the cell type. And if we focus on one regulatory site and ask this question, that which of the 3 genes overlapping this locus are likely to be correlated or controlled by this DHS? So is that gene 1 or gene 2 or gene 3? I think most of you would answer gene number 3 because whenever these DHS is open or accessible, the expression of the gene is high, and wherever it's closed, expression of the gene is low. So we basically can link DHS number 3 to gene number 3, and we may find other links between other pairs of DHS and gene. So basically, if we can find those links and have a test statistics that find the correlation between DHS activity and gene expression, then we can find which genes are likely to be affected in this process. We applied RegFM to 9 different autoimmune diseases, and basically autoimmune diseases are a group of more than 80 complex diseases driven by systematic or tissue-specific immunological attacks, and it includes multiple sclerosis, rheumatoid arthritis, celiac disease, among many others. Genome-wide association studies have been successful in identifying hundreds of loci associated to autoimmune diseases. And almost 8 or 9 years ago, multiple genetics consortia for the autoimmune diseases got together and designed a customized chip to densely genotype 185 genomic regions with the association to at least one of the 5 major autoimmune diseases. So this data set was ideal for our study because basically the dense genotyping of the disease-associated loci are available, and therefore we don't need to worry about the imputations. Also, as you can see, the same chip is used for all of the diseases, and therefore we can compare them across multiple diseases in a consistent way without worrying about the biases. Also, the genetic association data is publicly available. I'm showing you one example here, which is an association locus for the rheumatoid arthritis. There are 10 genes overlapping this region. By applying RegFM, we found that there are 5 DHS with at least 1 credible SNP located on them. And majority of the signal goes to a single DHS. And when we run the last stage of the model, we could identify our IRF8 gene as the relevant gene. And basically, there are experimental evidence for the role of IRF8 in rheumatoid arthritis. So basically, by just a computational analysis, we can predict the relevant genes. There are several examples of this kind in our publication. I'm just showing you one more example. Which is about the regions that are shared by multiple diseases. So some associated loci are shared by more than 1 disease, and always there's this question that is the mechanism underlying these diseases are the same or different. One example is a region on chromosome 6 that has association to 5 different autoimmune diseases. We applied RegFM to all of these 5 diseases and find that for 3 out of 5, RegFM identified the same regulatory site and same gene to be relevant, meaning that the underlying mechanism for this locus could be shared for these 3 autoimmune diseases. And when we applied RegFM to the other 2, IBD and celiac, we found that different DHS and different genes were predicted to be the relevant components in these loci, meaning that the mechanism could be different for IBD and celiac compared to the other 3 autoimmune diseases. So basically, RegFM can help us to understand if the biological mechanisms or regulatory mechanisms could be same or different across multiple diseases. Okay, to summarize, we have developed a model to identify gene regulatory mechanisms of complex diseases and applied that to 9 different autoimmune diseases. Going forward, I would like to consider this question that when there is like some common events for example, in the case of inflammatory bowel disease, that patients can develop cancer. So is the mechanism underlying the autoimmune diseases and the cancer could be shared or distinct? Of course, this is just an earlier stage of the project, so the data is not available. At the end, I would like to thank my collaborators, especially Dr. Pugh and Brutno, and also the team members of my lab, Alain and Eric, for development of TME Explorer package, and our collaborators at the Broad Institute and Yale University. Thank you. I'm happy to take any questions.